Hello and welcome at the GCP Mindset channel. Today we speak about a medical device topic again, and I have my guest with me, Dr. Andreas Beust. He wants to explain us a little bit more about the changes of the ISO14155 in regards to biostatistical requirements.
Hello Andreas, thanks for having me again.
Yes, Andy, thanks for being here to talk with me about the update of the ISO14155. And I think for the medical device world we have a quite tough time. First the MDR with all the new changes which will come very soon, and now an update of the ISO14155. And in particular in biostatistics we will expect a lot of changes to make data more robust. As far as I understood them. So now I'm no statistician. Please explain me what happened in the past. Were the data not reliable? Was there a lack of robustness in data?
Well, in particular, they were reliable with regards to sponsors that took a lot of care in planning their trials, but that required both a bit of experience as well as guidance by experienced, for example, biostatistician. Now, if you didn't have that, then the requirements were so thin in their complexity as far as they have been written down in the ISO14155 that a lot of concepts were left to the decisions of the sponsor on how they want to describe it. Nowadays, more and more requirements are set out to standardize the whole process, which probably also goes in line with the unification of the medical device sector with the pharmaceutical industry in terms of guidelines to just ensure that all of the data from both of these sectors are sort of generated in a way that yields reliable, robust data that also ensures trust of the public into the devices. And beforehand, there were just a lot of ways to circumvent having to actually produce robust and reliable data, which now there are a lot more details to ensure this is covered properly.
Okay, thanks, Andy. For me, it sounds a little bit scary. There's a huge variation of sponsors. Some plan the studies very well, have chosen a very good study design. Others don't do that and did not take care of biostatistical questions at all. Now the ISO 14155 will change, especially in terms of the biostatistical requirements. And I just want to discuss with you point by point what it means for the sponsors. Point 1 says, analysis population, for example, intention to treat per protocol as treated and procedures that take into account all the data. What does it mean for a medical device study sponsor in future?
Well, generally you have to consider that within a study, patients will be enrolled and they should follow the protocol from start to finish and eventually terminate the study. However, this is just not the case for all of the patients. And as much as you try to reduce factors that may lead to patients dropping out early, there will be some, and there will be some deviations from the protocol as well. And it needs to be pre-planned how to deal with these scenarios to ensure that the analysis that is performed afterwards is done in the most optimal way and in a way that is also not driven by knowledge of the data that is accrued within the trial. And therefore, these things should be set out beforehand in the protocol to clearly identify which populations will be analyzed and for which analyses they will be used.
Okay, that means that actually all patient data needs to be used in future and not only the best data which match according to the expectations best.
Exactly.
Yeah. Okay. Point B says then descriptive statistics of baseline data, treatments, primary and secondary endpoints and safety data, while point C adds analytical procedures including measures of precision such as a confidence interval if applicable. What does that mean?
Yeah, so nowadays in protocols and of course also in the appended statistical analysis plan, you should be very precise with which analyses you want to perform. And that basically means everything except for the exploratory endpoints should be predefined. And this includes just general summary statistics as well as more complex statistical analyses. This is very important also for the sponsor to know, since it means that when deciding, for example, which assessments are to be performed within a study, they should really get a good understanding of how these assessments will contribute to the overall study results, what information they can get from it, and which analysis they plan for it. This means that most of the analysis should be pre-planned, and then of course additional exploratory analysis can be added afterwards prior to, for example, closing the database. But they should take greater care and be more detailed in clearly setting out the envisioned analysis at the beginning of the clinical trial. That means when devising the clinical study protocol.
Okay, the next point is quite complicated, I would say, especially for people who are not biostatisticians or don't have that much knowledge in that topic. It speaks about the requirement in terms of significance levels, and it's very specific even. It says which significance level is required for particular studies, and even what kind of power and how the power level actually should be reached in particular studies. Could you explain it a little bit more in detail so that everybody can understand it?
Yes, of course. So in past times, usually there were no confirmatory analysis done, especially not prior to marketing a medical device. So this is something that is set out in more detail now to ensure that also those devices that are just trying to get a market approval are tested in a proper way. So therefore, the type 1 error rate, which means the likelihood of rejecting a null hypothesis, although it should not have been rejected, is very particularly set out here to ensure that there's a standardized way of doing it. Of course, there's some flexibility, which is also mentioned in this point. More important is also, of course, the power, and there are other points that target sample size calculations, which means the chance of detecting significant differences if there are any. So these need to be pre-specified just to ensure that there's a common ground for the analysis within the trial. Now, what's particularly interesting about this point is that multiplicity is mentioned in here as well, and that is also once more mentioned in point K of the amended ISO 14155. And this is an issue when you test several times. For example, if you have multiple endpoints, or if you test one endpoint several times consecutively, for example, during interim analyses. Now, the problem here is that if you test it once, you have, for example, a 5% chance of making committing a type 1 error, which is usually where these are set at for two-sided tests. If you test several times, then each time you can again make this error. So the error probability accumulates, and therefore the overall likelihood of you committing at least one of these errors is increased. And that needs to be controlled, and it needs to be described in detail whether this is an issue and how the issue is dealt with.
Okay, thanks, Andy. The next point is also very detailed. It's point E. It explains the requirements for sample size calculation. And the sample size calculation and justification taking into account all relevant clinical data on outcome variable and effect size, assumptions of expected outcomes across treatment groups, adjustments due to any pre-planned interim analysis, if applicable, detectable effect size, and if applicable non-inferiority margin, which should be smaller than effect size. 5, randomization allocation ratio. 6, expected dropout rate. And all these points should be taken into account during a sample size calculation. I think, according to my experience, that is really for many sponsors new, because most of them said it's more an exploratory study and we don't need an official sample size calculation. At least in the past it was the case.
Yes, indeed. A lot of the times no sample size was conducted, or at least no formal sample size calculation was done. So it was more of an estimation of how many patients may be required to perform the analysis, and to standardize sort of the quality of the outcomes that are generated by clinical trials. This now has a lot more emphasis put behind it. And therefore also this abstract is so detailed, because a lot of sponsors don't realize how many assumptions need to be made to come to a meaningful sample size. And therefore this is very detailed in setting out all of these parameters that should be considered. And the process of actually calculating the sample size, once all assumptions are there, is, let's say, relatively simple, because great tools exist for this. However, getting all of these assumptions and also having meaningful assumptions that are both scientifically and statistically sound, and that sort of yield a study design and a sample size that is good enough and gives enough assurance that also a successful trial can be conducted, is very crucial. Now, as always, sample size should be selected in a way that as few patients as possible are included, but as many as necessary. And here it should be, great care should be taken that the overall power of the study, even given results that are worse than what assumptions were made, is actually reflected within the sample size calculation.
Okay, the high level of details means more, it should be a helpful guideline, more a checklist instead of scaring the medical device sponsors.
Yes, indeed.
Yeah. Okay. Point F says description of how many procedures, if any, should be performed by a single user as part of the learning curve and how these data are to be analyzed. What does that mean? What is a user?
Yeah, so this aspect seems to be very particular for medical devices, since if you take a tablet, then none of the persons involved are really affected or need to be trained how to use the tablet. This is different if you have a medical device. Its implantation or its use by the patient or the investigator needs to be carefully trained first before you use it properly. At least for some devices that's the case. So when planning a trial, it needs to be considered whether training needs to be performed beforehand, and if so, then how many trainings need to be performed. And also the data that is generated during these trainings can be used, or maybe it may not be used. So this is something that should be set out in detail in the protocol, and the minimum requirement set out here is that at least you need to justify why you do not adjust for any learning curves within the protocol.
So that means training is also considered bias.
Yes, definitely, because if you have an unexperienced investigator, then he or she will likely result in, let's say, less optimal outcome from your medical device as compared to a trained one. So selection of investigators within a trial can definitely impact the outcome that you observe.
And that is also probably very important when you compare one device against another device, that the training is really standardized and can be compared.
Absolutely. This, of course, always depends on the device that you're looking at, but it should definitely be considered what is done, for example, for comparator devices, if they are included within a trial, and to standardize the process to get comparable data.
Okay, that's of course understandable. So therefore, let's jump to the items I and J, which say something about management of bias and confounding factors, which can include randomization, matching, blinding, stratification, and stratified randomization. What exactly does that mean?
Well, there are two things here. Bias, first of all, is sort of the concept of having a design choice that fundamentally influences the outcome of the trial in any way, shape, or form, so that it deviates from the actual sort of underlying outcome. So, for example, you get a more positive or more negative outcome simply based on the design, because it sort of shapes the results in a certain way. And a good example of this is if, for example, an investigator were to rate the probability of a patient successfully conducting the trial and having a positive outcome, and then deciding whether that patient should be allocated to the treatment arm or comparator arm. So that way he could allocate all of the promising candidates into the treatment arm and all the not so promising ones in the comparator arm, and then you would immediately have a bias because the results would deviate from what would factually be observed within the trial. And so therefore, a lot of these bullet points mentioned here within the guideline aim at reducing bias systematically within a trial by predefining these things and ensuring that at less points as possible, few points as possible, there is the opportunity to modulate the trial outcome. Now, confounding variables are those that impact, statistically speaking, both the dependent and independent variables. If you, for example, take a study that tries to look at whether alcohol consumption influences mortality, then these are your dependent and independent variables. And those are both affected by the age, for example, of the trial participant, or its sex, or its education. So if you may, after the trial, identify a tendency that those that consume more alcohol have a higher mortality, but both of them may also be more predominantly males, which generally drink more and have a higher mortality, or of lower education. So identifying these factors that can impact your results is very important, and this needs to be described also in detail, prospectively in the protocol.
Okay. Could you shortly also explain what's the difference between blinding and masking?
Well, generally, masking and blinding are the same. It's just that the term blinding is, of course, negatively associated, for example, in ophthalmologic trials, where you implant lens into the eye of the subject, so you wouldn't want to describe the study as a blinded study.
Okay, that's a typical term then just for eye care studies.
Generally, yes.
Yeah. Okay, understood. Let's go one point back, which actually did not change. Let's speak about interim analysis, and especially in the medical device world, where companies are usually smaller. There are a lot of very curious people who want to know very early the results of the study. But most of them don't know that interim analysis also might have an impact on sample size. Could you explain a little bit why this is the case?
Yes, so performing an interim analysis can come with two issues. One is bias and one is multiplicity. So as far as the bias is concerned, having an early look at the data and having an early evaluation of the data will mean that any changes performed afterwards are likely driven by knowledge of that data. If you have really good results, then maybe you overestimate the outcome that you will possibly receive at the end of the trial and make decisions based on that. Or if you have actually quite bad results or results that do not fit your expectations at the interim look, then you may change the trial, may even terminate it early, simply because you're afraid that you are wasting time and money. So this is generally a very big issue with performing interim analyses early. Although it's of course very comprehensible that medical device manufacturers are of course interested in early looks in the data. The other issue that you mentioned is if you already perform tests for the primary endpoint, for example, and you already compare maybe treatments, you do assessments with it, and you try to get to a conclusion, then you run into the multiplicity issue. That means every interim analysis and every look that you have means that you can commit a type 1 error. And this of course accumulates then if you have several interim looks, so that in the end your overall type 1 error is higher than what you initially set it out to be. That means 5% for the overall study. So to adjust for this, you will need to reduce the type 1 error allocated to each of the analyses, which includes also the final analysis. And that can yield to an increased sample size because the type 1 error probability also determines of how many patients you need to ensure that significant differences can actually be shown.
Okay, very good. So let's continue with point L. The specification of subgroups for analysis, if applicable, or if response to treatment is expected to be different in these groups. What does that mean?
Well, basically you have to consider that studies should generally try to be as broad as possible to allow for applying these results that were gained to a broader population that will later on be treated. So there should be a sense of generalizability of the results. But that yields itself to different factors influencing the results that you maybe didn't control for. And therefore, it's been specified to greater detail in the updated ISO14155 that the sponsor should really take care of identifying factors that may influence the outcome before. We have talked, for example, about the experience of the investigator before, and this is something which can be investigated, whether different study centers within the trial amount to different results overall, and therefore whether the training of the investigator may have an impact on the outcome, which is of course very important. If you want to later on sell this device, then maybe you need to also sell the training with it if it's required. And the same thing can be said, for example, for different age groups or different predispositions within the trial. So it just needs to be thought about very carefully which factors may influence the outcome and whether smaller groups sort of should be formed and then analysis performed on these to identify differences between them.
Okay, thank you very much, Andy. It was very nice that you explained it so easily for us. But in general, I'm wondering how the medical device industry can cover all these changes. And I mean, in general, the companies are small. Sometimes they don't have medical doctors involved, sometimes they don't have scientists, sometimes they are just engineers, which makes perfectly sense. But now I think in future they also need a lot of biostatistical consultancy, and not only a biostatistician from the academic world. I think pragmatic biostatisticians are very important. Otherwise, they need to run such big studies like the drug world, and that is not the new requirement. I mean, you still can run smaller studies compared to the pharmaceutical industry, correct?
Yes, of course. Also in future, medical device trials will be smaller than those run by the pharmaceutical industry. Although the requirements set out for these trials are increasing and sponsors really need to ensure that data is gathered in a way that the analysis yields robust results, it still doesn't mean that the sample size needs to explode. It's just that the trial planning needs to be conducted in a way that really makes sense. And it's also important to keep in mind here that the most expensive study is always the one that fails to show what you want to show, either by not achieving the necessary requirements to reject the null hypothesis of the primary endpoint, or by generating meaningless results and then being forced into repeating the trial afterwards. So it's just that the sponsors need to take good care that they consider all of these statistical aspects also very early on in the trial, that they seek advice if they do not have the capacity. And as you said, a lot of the medical device manufacturers are small businesses. And therefore, it's important that they simply seek these advices to optimize their planning and ensure that in the end, the trials also run in a way that doesn't yield extremely high costs, which would sort of prevent them from ever entering the market.
Andy, great. Thank you very much. It was very helpful, and I really hope that medical device companies like such videos and they motivate them to ask you before planning a study, and that they are not scared of the changes but see you more, or guys like you, more as a helpful guide to run the study more effectively in future.
Yeah, let's really hope so, because that's the only way of regaining also the trust that some of the medical device industry has lost, unfortunately, over the years.
Yeah, very good. So thank you very much for watching us. Please send us your comments and questions, of course, and recommendations for next videos, and see you the next time. Bye-bye.
Bye.